Okay, I think we should start the class. So I'll be sharing my screen. Please let me know whether the screen is visible and whether my voice is clearly audible or not. Anyone?
It's visible, sir, and
Okay, okay. I think
you're also audible.
okay, okay. Thank you. Thank you so much. Okay. So first of all, a very happy Holi to you all. So hopefully you had a nice celebration at your home or in your institute, wherever you are. So again, with that, I welcome you all again to the live tutorial session for week 8's content for the course Bioinformatics Algorithms and Applications, instructed by Professor Michael Gromiha from IIT Madras. So from the weeks in week 8's contents, basically Professor Gromiha, he went over several terms and topics and related discussions on, for example, hydrophobicity, surrounding hydrophobicity, mutations, and how those might stabilize or destabilize certain regions of the protein based on those surrounding environments, and different basically numeric concepts, numeric problems related to those as well, right? Excuse me. So we'll look at that and we'll try to solve some similar questions which also came in some of the earlier iteration of these courses, similar to what you might be getting in your assignments as well and your exam. So in the past, I think a couple of classes we went over, for example, the just like as a quick recap, so you went over the PDB structures, the different components of that, and in the latest class, I think we discussed about homology modeling, right? So and along with that, I think we solved some of the numeric problems. So I thought we should start probably this class with a similar type of numeric question that I wanted to kind of discuss in the earlier class, but couldn't. So say you have been given like a PDB structure, and then of course the PDB structure will contain. If you look at the information inside, so it will contain the residue information, right? Informations for all of the residues, and each of the atom that the residues consist of, it will contain their x, y, and z coordinates, right? Now, based on this x, y, and z coordinates, what you can do also is that you can measure distance between different atoms, right? And those sort of distances might give you an idea about, let's say, certain changes that is going on in the structures. If you let's say simulate them, so you might be able to observe those distances and how they change over time. So this kind of analysis is also quite uh, done quite a lot in different computational basically characterization of proteins and protein dynamics and so on. So where you have to basically calculate the distances between two sets of residues, or it could be two residues. It could be the center of mass between the different sets of residues. It could be center of mass between, for example, ligand and some protein residues, right? So different sort of atomic distances or center of mass distances of the residues that you might need to calculate in order to get some structural insights into basically the dynamics of the protein or something else. So in this particular question, similar type of, for example, answer is being asked. So you have been provided a certain part of the PDB structure, right? And this is the structure of a protein. And what you have been asked to do is that, so these are basically cysteine residues. So this is only uh, the sulfur atom for certain cysteine residues that are present in the protein, in the PDB structure. Okay. So only those atoms informations have been given here. Of course, we know what all of this column means, and in this particular portion, if you carefully observe, we will identify that, okay, these are the x, y, and z coordinates for the said atoms. Okay. Uh, so different cysteines are present in this particular, in this particular structure. So we have 1623, we have 1697, 138, 192, and 236, right? Now, basically, if you know, cysteine residues, they can form disulfide bond with other cysteine residues, right? And together they can kind of become a cysteine. Okay. So these disulfide bonds are also important in a way that basically it can help stabilize some portions of the protein via these disulfide bonds. So in this particular question, it is being asked that, okay, if you have been given all of these cysteine residues and their information, so which of these cysteine residues will be forming disulfide bond together? So what you have to basically do is that you have to calculate pairwise distance between two cysteine residues. So you have to kind of take all of the pairs and try to calculate the distances between these atoms, the SG sulfur atoms. And then if you notice uh, any sort of pair of residues which are in disulfide bond forming distances, so then you can say that, okay, these two cysteine residues are probably forming disulfide bonds, right? So that is what being asked, what is being asked in the question. So could you please maybe try to calculate some of these distances and share in the chat which uh, cysteine pairs could form disulfide linkages in these among these. So for disulfide bond distance cutoff, you can think of it like an average 2.3 angstrom, something like that, or maybe maybe a little bit higher or lower. Obviously, the cutoff should be higher a little bit maybe. So you can take around that distance as a cutoff for disulfide bonds. And what you have to do basically is that you have to calculate the distances between these atoms and then try to figure out which ones could be forming disulfide bonds. Right. And for the formula for calculating distances in a 3D space between two coordinates, two sets of points, you can use this one. So x2 minus x1 whole square plus y2 minus y1 whole square plus z2 minus z1 whole square. You can do this summation and then you have to take a square root of that, right? So for example, here x between two, for example, atoms. So x2 and x1 will be their the difference between their x coordinate values, right? So with that, which cysteine pairs could form disulfide bonds between these uh, among these which are given. So for example, if we take a look at the distance between 23 and 97, of course, you will notice that if you put all the values, like for example, minus 14.47 plus 12.77, and then you do the square of that, right? Uh, do that for all of the x's, y's, and z's, and then you take a square root. So ultimately, you'll be getting something like this: 2.046 angstrom. So which is in disulfide bond distance range, right? So then you can say that 2397, these two uh, cysteine residues are forming disulfide bonds, right? So then any other sort of cysteines that you think are forming disulfide bond that are present in this list? So if you notice, okay, 138 and 192, I see one answer in the chat. Yeah, in this case, the distances between residues 138 and 192, the sulfur atoms of these cysteines. If you do the calculation, you'll find out that it is around 2.051 angstrom. So yeah, of course, they're forming disulfide bonds, right? So these type of like numeric problems where you have been asked to calculate certain distances in 3D space, uh, these could be important, right? In your exam and assignments as well. And in research processes also, usually you don't have to do these calculations by yourself, of course, because usually you will give certain software some commands and they will calculate the distances for you. But the calculation of different distances is always an important part of computational analysis, to say the very least, right? So that is why we need to also understand the mathematics of formula behind those as well. So yeah, in this case, the answer would have been 162397, that pair, and then 16138 and 16192, that pair, right? Okay. So in the earlier classes, when we, of course, talked about different statistical parameters that can allow us to assess the deviation of different structures, right? The deviation between different structures of the same protein, let's say. So we came to know about one particular parameter which is called root mean square deviation, right? Its formula was something like this. For example, RMST mass weighted. So you had to sort of calculate, for example, the particular atom I. You had to do uh, basically calculate the x, y, and z points deviation from a reference atom, different reference atom, which is in this case particularly it's talking about yi. And then for the x, y, and z coordinates, you have to do that. Uh, and if it is not mass weighted, then you have to just divide by the total number of calculations here, the number of atoms ultimately in the end, and you have to take a kind of square root after the summation, after the whole process basically. So RMST can give you an idea about structural deviation or conformational changes that is occurring in the whole protein, it could be, or in the certain parts of the protein. But in the earlier classes, we also discussed about protein folding, right? So protein folding in the sense that we discussed different concepts regarding that. Uh, for example, from unfolded to folded structures, we also tried to calculate the folding free energies. Those sort of questions were also there. Uh, and then we also, you know, learned about the prediction algorithms uh, for, protein folding, basically, which try to predict the folding state or folded state of its protein structure, right? Now, to investigate protein folding, or let's say the change of folding from unfolded to folded state, or maybe it is already folded in a 3D structure, but let's say due to the movement of different loop regions of a protein, so inward movement, outward movement, basically collapse or uh, extended states of those proteins. So that will also change the like overall nature of the fold, right? And one particular statistical parameter that is always used, that can be used in basically characterize the protein folding, whether there is any significant change between the folded and unfolded, between different states or not in terms of protein folding. Uh, and also it can basically allow you to assess the compactness of a structure overall. So does anybody know any such kind of statistical parameter that is widely used in molecular dynamics analysis also, to sort of assess the structural compactness, and which can also give us an idea about the change in the fold of the protein, or along those lines. So does anybody have any idea about this particular statistical parameter that I'm trying to mention? So I thought I would, yeah, please. I think I saw one hand.
Sir, I don't know exactly, but correlation.
So correlation, I guess, maybe between different parameters. If you are trying to correlate, that could give you an idea about the change in the folded state. So correlation is way too broad, I guess. But yeah, so in this particular one, which I was, the parameter that I was trying to mention is radius of gyration. So radius of gyration is basically a parameter that lends us insight into the structural compactness. And if you compare, for example, a protein in its unfolded state to a protein in its folded state, so of course the folded state will be more compact, right? So in unfolded state, all the loops will not have been probably clumped together. So all the basically the primary structure and the secondary structures are not sort of clumped together in a small sort of space, right? In its folded state, that sort of compactness parameter changes, and it essentially becomes more compact. And even in the case of, for example, certain loop moving outwards or inwards, that can also change the overall compactness of the protein, right? So that is, those kind of things can be basically characterized by this parameter, which is called radius of gyration. So since we learned about the different statistical parameters, I thought I would also include this uh, basically briefly in a slide. Uh, so firstly, to understand radius of gyration, for example, we have to understand moment of inertia, right? Now, inertia, as we know, is the tendency of a particular particle to stay in a rested state or in a motion state, right? And this is, dealt with in Newton's first law. So, moment of inertia is basically the same concept, but it is regarding the rotation, as it is mentioned here. So, it is basically the property of a body due to which it opposes any change in its state of rest or uniform rotation. Now, if we have, let's say, just bring this entire kind of slide. Uh, so if we have a mass m here, which from a certain axis of rotation has a distance of r, then we could express the moment of inertia I equals mr square, right? For a single mass of m, which is at a distance r from a axis of rotation. Now, if there are, let's say, multiple masses which are at different distances from the axis of rotation, then we could basically use the formula summation of mi ri square to calculate the moment of inertia. Now, the radius of gyration is basically takes that concept from the moment of inertia. So it is basically saying that uh, radius of gyration around a specified axis, so it represents the perpendicular distance from that axis to a point where if the entire mass of the body could be concentrated, that would not alter the moment of inertia of the body around that axis. Uh, in terms of the formula, if we write it down, so it would be I equals basically m times rg square. So this rg will be the radius of gyration essentially. So for example, in a complex system or a complicated system, we had to do this sort of summation, right? Summation of mi times ri whole square. And of course, if you take mi as common, uh, then what you are getting essentially for the radius of gyration is a representation of all of the distances of all of the particles that the system is constituting of from the axis of rotation. So if you convert, if you write Rg in terms of moment of inertia and the total mass, so it will be root over of I divided by m. And for all of the constituting particles, their distance from the axis of rotation, if you want to write the radius of gyration in that term as well, then you can use this formula. So root over of, for example, inside you have r1 square plus r2 square plus r3 square up to rn square divided by the total mass. So this is the radius of gyration, which you can use, again, a parameter which you can use to monitor basically the change in the folded nature, or for example how much percent the protein is being folded, or if it is moving toward a folded state or an unfolded state, such kind of things. So maybe I would just uh, show the plot for a second. So if I share it, so hopefully this plot is visible. So this is something that I just wanted to include because it is also another interesting parameter which can lend us insight into the folded nature of the proteins, right? So here, what we basically have. So it is basically the parameter files or the files for analyzing a molecular dynamics trajectory. So whenever you are trying to analyze a simulated trajectory, you first of all need a topology file which uh, sort of basically describes the relationship between all of the atoms in that system in a topological form. So that will be the topology file kind of, you can think of it that way. And then you have to load the simulated trajectories. Uh, and then here, if we take a particular look for this command, so this is the command for the particular software suite, which is called Amber. So Amber has an analysis module, and it is kind of a part of that. So here, what we are basically doing is that from 1 to 277 residues of a certain protein, we are taking the C alpha atoms, which is represented by the Ca, and we are calculating the radius of gyration of that. Okay, and we are using we are basically appending all of the data to this output file, radius of gyration underscore Ca dot that, because it is for C alpha. So after running, for example, this analysis, when we get the result, if we try to do, for example, a simple line plot for the time series or how the time evolution of the radius of gyration looks like. So in this particular case, it looks let's say something like this. Okay. Now, of course, as I said, that it can lend us insight into the folded nature of a protein, because the radius of gyration is essentially related to the structural compactness. If we had a not so compact structure, right? You can imagine its radius of gyration would be larger. Okay, and if you had a very highly compact structure uh, in a highly folded state, uh, then that will be having a radius of gyration of much lower value. In this particular case, for example, if we uh, try to look at the result, we will see that the radius of gyration value is decreasing. Let's say from the initial time point, and then after a certain point it kind of becomes stabilized, right? So this sort of analysis might lend us insight into the uh, uh, it can basically tell us that okay, since the radiation direction is changing from the initial state to, let's say, right up till this point it was kind of declining, right? It is not that much. Let's say in this case it is only 19.6 to 19, so zero point 0.6 change in the value, but let's say the change was in a much larger amount, right? So let's say if it was changing uh, in like 4 or 5 angstrom range, then we can say, okay, so there might be some significant change in the compactness of the protein happening, which can basically give us an idea that, okay, some sort of change in the fold of the protein might be occurring. So it could either be, some loop moving inwards and outwards, or the change in the fold of the protein, these kinds of things. So this can give us basically an idea of that. So this is just something I wanted to uh, briefly include. Okay. Again, I will share the other screen. And yeah, so now we'll move towards some of the concepts that Professor Gomiha actually went over in his classes. Uh, and in the uh, basically lectures of or the contents of week 8, so we learned about different concepts regarding, as I said, hydrophobicity, surrounding hydrophobicity, mutation, their impact on the stabilization or destabilization, and so on. So these things, right? So surrounding hydrophobicity is one concept that we, of course, learned about. So what it is basically uh, characteristics. So this is one of the characteristics that is considered while looking for stabilizing residues. So what surrounding hydrophobicity tells us, or the way to calculate it, it is basically that you have to, for a certain residue, you have to take, let's say, distance cutoff. In this particular case, it is taken as 8 angstrom, and it is for the threonine, C alpha atom of threonine, and then you have to kind of imagine a sphere in this region, right? And whichever residues that is sort of falling within that sphere, within that bubble, for this 8 angstrom cutoff radius, those residues will be considered as part of the calculation of the surrounding hydrophobicity. Surrounding hydrophobicity is basically what type of environment in the surrounding basically region of this particular residue do we have in terms of their hydrophobic properties, right? And the formula to calculate that is basically hi equals to summation of nij times hi, small hi. So in this case, hi, capital Hi, this will be the surrounding hydrophobicity of the central residue J. So we are trying to calculate the surrounding hydrophobicity for that particular residue. And then Nij and Hi. So Nij is basically the number of residues of type I around J. So imagine there are all sorts of different types of residues around this particular threonine here, right? So there could be multiple of the same residues. For example, let's say 4 leucines are present within that sort of sphere, right? Within that cutoff radius. So then in this particular case, the number of residues of type I, so in this case type I could be, let's say leucine. So that you have to consider around this particular threonine, the residue J, and somehow you have to include the experimental hydrophobicity of the residue I, which in this case, if you consider, let's say 4 leucines are present within that radius of 8 angstrom around threonine, then we have to include the experimental hydrophobicity of this leucine as well in this calculation. And ultimately, you just do a summation of all of that, and in total, you get the surrounding hydrophobicity value, Hi value for particular residue that you're trying to calculate. So here, Professor Gomiha, I think, mentioned in his slides some values taken from databases. So for different residues, let's say here, the surrounding hydrophobicity values are mentioned here. Okay. So of course, these indicate what sort of hydrophobic environment does these residues reside in. Okay, so that's just basically a way to quantify the environment, the nature of the environment. So apart from surrounding hydrophobicity, Professor Gomiha went over some other terms also. For example, long range order. Now, of course, long range order relates to long range contact. I think it is written wrong here, but so long range order, of course, relates to long range contact. So does anybody remember what long range contact was about, and what was the other on the counterpart, if you will, short range contacts? So the difference between basically long range and short range contacts, does anybody remember? So it was basically relating to the residues that are forming contacts in the 3D space. And those contact forming residues, how far in the sequence space are those situated in? So long range contact between amino acids, 8 residues apart. Yeah, so basically that is a cutoff of 8 residues. So let's say you have to first of all take a cutoff for residues forming contact in the 3D space. So you have to, let's say, take some certain angstrom cutoff. It could be 7, 8 angstrom, something like that. So if certain residues are within that range, that particular angstrom range, you can say, okay, they are forming contacts, right? And then long range contact will be again another set of cutoff you have to take, that these contact forming residues, whether they are present close by when you consider the sequences, or whether they are present like far apart if you consider the sequences. So for example, if we have let's say a valine 2, let's say, and let's say leucine 5, right? So valine 2, leucine 5, they are forming contacts in the 3D space. Since they are only, let's say, 3 residues apart, so we can say that, okay, they are forming short range contacts. Let's say the cutoff for the long range versus short range contact was, let's say, 8 residues, as you said, if we take that as a cutoff. So that will mean that this valine 2 and then the leucine 5 or leucine 6, these are forming short range contacts, right? And then let's say another leucine that was present at, let's say, residue number 25, right? So valine 2 and leucine 25, these two residues are also forming contact in the 3D space. That means that in the 3D space, they are, let's say, within a range of 8 angstrom from each other. But in the sequence, if we look at it, so valine is present at residue number 2, the residue position 2, and the leucine is present at residue position 25. So since they are such far apart in the sequence, we can say that they are forming long range contact, right? And basically, Professor Gomiha has introduced, so the formula for calculating the long range order, so it will be something like this. So let's say we can say long range order equals uh, summation of nij divided by n. Now, in this particular case, this nij, it will be kind of a binary classification. Okay. So if two residues are forming long range contacts, then we can probably count it as this nij value as 1. If these residues are not forming long range contacts, then we can write a 0 for those. So some sort of conditions like this can be included, right? Nij equals to 1 if uh, the distance between I and j, let's say something like this, we write. So let's say we use 12 angstrom as a cutoff. So if it is greater than 12, then we will write nij equals 1 if we use that cutoff, right? And conversely, we will have to say nij equals 0 otherwise. So this is sort of a way to again quantify uh, the long range contact basically in a protein. So in this particular case, if the I and j residues are, let's say, more than 12 angstrom apart, then we can write as 1 for the nij value. Otherwise, we will write 0 for the nij value. And we can do the summation and divide by the total number of residues, and that will give us the idea about the long range contact, right? Sorry, the long range order. Okay. So you can imagine that you can use that to uh, basically characterize whether like how many long range contacts are being formed by certain residues, and you can compare and contrast between different residues and so on, right? So that would be useful. Another thing that Professor Blumenthal went over was stabilization center. Now, stabilization center, if we take a look at it, so I thought I would just take a look at the one of the original papers that sort of discussed about the stabilization centers. So this paper was kind of published in 1997 in Journal of Molecular Biology, and it is talking about the stabilization center in proteins, their identification, characterizations, and predictions, right? And in a certain part, we'll see that it sort of gives some definitions about the stabilization center. So first of all, it's talking about that, okay, two residues are considered to be in long range interaction if they're separated by at least 10 residues in the sequence space. So in this particular one, in this particular paper, they are uh, they are considering the cutoff that we earlier, for example, used 8 or 12. So in this case, they are counting as they are considering it as 10. So if residues are more than 10 spaces apart, then we can say that they are forming long range contacts if they are forming contact in the 3D space. Okay. And then what it is talking about basically is that residue clusters are identified in protein contact maps where an accumulation of long range interactions are observed. So this type of long range uh, interactions via the long range contact analysis, if we take a look at them, then we might be able to see different clusters that are being formed in a contact map. So contact map, you can think of it as like a map or a visual way of representing which residues are forming contacts between each other. Okay. So among those clusters, if we analyze uh, those clusters will contain the residues as cores, which can be called as stabilization centers. So these cores or these clusters, in their cores, basically the residues that are situated there, they can be called as stabilization centers, stabilization center residues. because you can think of it in a way that uh, they could have some sort of role in the stability of the 3D structure, right? Because you can imagine, from long range contact analysis also, so if a particular residue is forming a lot of long range contacts with different uh, other residues that are present in the protein structure in the 3D fold. So you can imagine that these particular residues are, of course, crucial member or crucial part of the stabilization of that particular region or this particular structure of the protein, right? Okay, so you can imagine that these sort of residues are very crucial in the interaction, in uh, basically providing stability to the structure of the particular part of the protein. So that is kind of the uh, basically formal way of describing what the stabilization centers could be. So you can take a look at this paper maybe for other concepts regarding those. And another concept Professor Blumenthal went over was conservation score. Now, conservation score, as we also discussed in some other classes as well, that it basically talks about, let's say, in a sequence, uh, at a certain position of the sequence, are the amino acid residues conserved or not? So, conserved it could be in terms of, let's say, the same type of residue that are present at those positions. So, conservation uh, based on their, for example, hydrophobicity polarity as well, or let's say the same residues are we are just considering as uh, analysis for this conservation scores. So it could be that if let's say in a sequence at position 3 a valine was present, and then after different sequence alignments and so on, we are seeing that in a lot of other similar proteins for the position 3 the valine remains the same, then we can say okay, for position 3 valine is a conserved residue, right? So similarly, if you talk about not only a single residue, but let's say certain parts of the proteins, you might be able to identify some sort of structural motifs or which are basically conserved across different species and so on, right? For example, if we talk about the protein kinases, you will notice a lot of conserved regions that are present across different kinases, right? In humans, in other animals, in other species as well, and so on, right? Uh, so those that is basically the importance of conservation score and the idea of basically calculating the or quantifying the conservation of certain residues at certain positions in the entire sequence or in the structure of the proteins, basically. Okay, so if we talk about some of the numeric questions that could be related to the concepts of stability, mutation, and so on. So similar questions might also appear in your assignment. In this particular one, it is asking that it is giving you basically 4 different mutations, which all are valent to alanine, but basically at different positions, these valines are present. So valent 12 to alanine, 24 to alanine, valent 25 to alanine, and valent 28 to alanine. For all of these mutations, you have been given certain free energy change values. So 1.1 kcal per mole, 1.1 again, minus 0.1 kcal per mole, and 0.3 kcal per mole. Let's say for these 4 different changes, right? 4 different mutations basically. And in this case, we can assume that uh, the mutation, let's say for the positive free energy change could be, let's say, uh, destabilizing. It could be. We can assume that in this case, since the notation is not given, but that's not important here. Uh, so it is asking that based on the average assignment method, what could be the free energy change of a different valent to alanine mutation, which is present at position 35 basically. So valent 35 to alanine, if that valent was to mutate, what could be the free energy change of that particular mutation? And it is asking us to calculate it based on this particular method, which is called average assignment method. So could you maybe try to calculate and share in the chat? So basically, since it is talking about average, uh, then of course it uh, will require us to consider all of the different similar mutations. Means all of these similar mutations that have already been observed uh, in other residue positions, let's say. and what kind of free energy changes those valine to alanine mutations brought about. And based on this method, this average assignment method, we have to calculate. So maybe you could try to calculate it out and share the answer in the chat. Okay, so I see one answer 0.6. Yeah. So as the name of the method suggests, so it is an average assignment, right? So that means that since it is a valine to alanine mutation, so we have to consider all of the valine to alanine mutations for which the free energy values are observed or given, and then we have to take basically an average of them, right? So average in this particular case is basically mean value, right? So you can take all of the xi values here. You can do a summation of all of that and divide by the total number of uh, observed data points, right? Total number of data points that we have basically. So if you calculate the mean, it will be, of course, if you just add all of this up, you will get 2.4. You divide by the total number of data points, so divided by 4, so it will be 0.6, right? And at the same time, alongside mean, what you can also do is you can compute the standard deviation. So standard deviation will be for each particular data point, which is xi, you have to subtract the mean value, take the square uh, value of that, and then you have to do a summation, and then ultimately divide by n minus 1, which is in this particular case will be 3, right? So if you do this calculation as well for each of the data points, for example, 1.1 minus, which was the mean value 0.6, that square plus 1.1 minus 0.6 square plus minus 0.1 minus 0.6 whole square, and so if you add all of these up, you will get something like, so if you do the entire calculation, basically you will get something like 0.36, right? I've repeated the 2 steps here, but anyway, 0.36 should be the standard deviation basically, which should accompany the mean value. So it will be kind of like 0.6 plus minus 0.36. So this question I think also appeared in one of the earlier iteration of the course, and they sort of gave an answer in a range between 0.55 to 0.65. I guess they would accept basically any answer between this range maybe. So this sort of way they provided the answer. But anyway, in this particular case, uh, the mean value was 0.6, which you can also see that it is falling just between these two values that they have given as range. They had given as range, basically, right? So these sort of numeric questions also might appear in the exams as well. Now for the average assignment methods, so the amino acid substitutions were divided into how many beans? Does anybody know? So since this is talking about the amino acid substitutions, so amino acid substitutions means we have to calculate sort of pairwise substitutions, right? So for each of the amino acids that are present, let's say 20 naturally occurring amino acids, so each of them, if we try to substitute them to a different amino acid, so each of them can be mutated into 19 different uh, other amino acids, right? So in this particular case, what you can basically uh, get the size of the total beans as, since there are 20 amino acids naturally occurring, and each of them can be mutated 19 different ways. So the total number of beans could be 20 times 19, which should be 38. Okay. Now for the next question, yeah, I think I see one hand in the chat.
Sir,
Mhm.
what is bins in that? Uh,
So when you talk about beans in different contexts, so in this particular case, it is basically how many single amino acid to single amino acid mutations that you are being able to observe. So that is the total number of beans in this case. In a common way, if we talk about beans, so let's say you have a large number of data points, right? And let's say the data point for the data set, let's say it's an XY data point, something like that, that has two values for each individual data point. Let's say there is a large range or large space that the entire set of data points cover. So what you can do is that you can divide that data point into small subgroups, basically. So let's say you are taking, so let's say if the entire span of the data set was like this, so this big, right? You can take a small range and you can uh, take it as a single bean, and then you can take similar other beans that cover the entire span of the data set, and you can see, okay, how many data points are falling within this particular bean, and then you can analyze that, and you can try to get the idea, okay, which of the small ranges that I took from the entire space that the data covers, so which of the small ranges could be the most populated bean. So this sort of analysis you can do. In this particular case, we are not dividing the total number of data points into some broad clusters. But in this one, we are basically taking each individual uh, sort of amino acid to amino acid transition as a single sort of bean, essentially. So since there are a total of 20 amino acids that can be mutated, each of them can be mutated 19 different ways. So you can think of it as a way that you have total of 20 times 19, let's say, category of values that you can put your data in. So in that sense, the bins are being taken as 380 in the average assignment. So basically, how many total boxes that you have where you can put your data, essentially, and each of these boxes in this case represents a certain mutation. So from one amino acid to another amino acid mutation, each of these boxes represent that. You can think of it that way. For each mutation, you might have like thousands of data. So those particular thousands of data for one box, it's a valine to alanine mutation. So we have to put all of those data in that box, right? So that is basically what is being referred to as the bean here. In other data analysis, you take basically from the entire span of the data set, you take small ranges and you consider that as beans, and then you try to decide, okay, which of these beans are the most populated, and that could give you some insight regarding the data distribution, basically.
okay, sir. Thank you.
Yeah, yeah. So that is kind of the concept of beans here. So for the next question, the question was asking that normalized B factors, the positive value of that are predominantly observed in stabilizing residues, whether the statement is true or false. Now we learned about B factors in some of the earlier lectures also, right? Professor Gomiha mentioned it in detail. That the B factors are basically indicative of thermal fluctuations that are occurring in the different atoms for a crystallographic structure, let's say, right? So if you open up a certain PDB structure uh, in a text format, basically, uh, from one of the column you will see that indicates the B factor value for each atom. And if the B factor value is sort of very low, let's say it is around, let's say 10, 20, something like that, then you can say, okay, those particular atoms are more rigid in a sense. And if the B factor value was high, let's say 75, 80, something like that, then you could say, okay, that is a high B factor. So those particular atoms are highly flexible. So there is much thermal fluctuation going on in those atoms, which can give us an idea about the flexibility of different regions as well. In this particular case, it is asking about normalized B factors. So the B factors have been normalized, and the formula for kind of calculating that normalized B factor would be, so if we, If we take this as equation here, but anyways, so yeah, if we basically calculate the B norm, so it will be B minus B mean divided by B sigma. So mean value, of course, we know that is the average value, and then the sigma in this case will be the standard deviation, right? And if a B normalized is greater than 1, then we can basically call it as flexible. And if the B normalized value is less than 1, then we can consider it as rigid, right? So in this particular case, the answer would have been false. We see a slide, for example, Professor Komiha also introduced or also mentioned in his classes uh, that the normalized B factors, if they are having highly positive values, uh, those will usually be indicative of flexibility, high flexibility, right? In this particular table, you can see that these negative values for the normalized p factors will indicate that those could be, uh, stable residues. But for positive normalized b factors, those will not be the stabilizing residues. Those will mainly cause destabilization of the structure. That's it, right? Destabilization on the structure, I mean, the residues will be highly flexible in that part. Okay. Now, similar concept regarding stability, if we talk about, so of course, the different regions of a protein, for example, the inner core of a protein or the surface regions of a protein, right? Whether that part is solvent exposed or is buried much deeper into the core structure. Uh, so those sort of uh, parts and what the surrounding uh, basically environment is and what sort of impact that could be observed by mutation, different types of mutations on those certain residues. So these kind of concepts can also be analyzed, right? So here in this question, it is asking that positively and negatively charged residues that take a predominant part over the hydrophobic residues in stabilizing a protein. Now, in this particular case, this stabilizing the protein, it particularly is talking about the stabilization of the 3D fold, basically. And as we discussed in some of our earlier classes, that the predominant driver of the 3D folding in protein, that was basically the hydrophobic clustering, right? So hydrophobic forces, because the hydrophobic forces in an entropy driven way basically clumped together to give the core part of the protein structure, right, which are away from the solvent. So in this particular case, again, the answer would have been false, because positive and negatively charged residues, they do not take part in the uh, they do not take the prominent part in stabilizing the 3D fold. They do stabilize the 3D fold in certain way, because positive and negative charge, again, if they are let's say forming electrostatic interactions and so on, so they can stabilize certain parts of the protein. But for stabilizing the entire fold of the protein, the major role goes to the hydrophobic residues, right? And you can imagine, so different types of mutations in the hydrophobic environments of the proteins. So if those mutations let's say occurred in a way that a hydrophobic residue was mutated to a charged residue. So of course that will cause a destabilization in this part, because it is not supposed to, a charged residue is not supposed to be at the core part in a hydrophobic environment of a protein, right? So that could be causing the destabilization of the structure. Again, similarly, if you see, let's say in a surface uh, region of a protein, so there should be, let's say, charged or polar residues that are present in the surface region, right? So if those charged or polar residues were to be mutated to a hydrophobic residue, highly hydrophobic residue, then you could imagine that since that will not be favored, uh, because the hydrophobic residue will not want to basically stay in a solvent exposed area. So again, that will destabilize that part of the structure as well. So these different type of mutations based on their surrounding environments and their preferences can cause structural destabilization, or conversely, they can also stabilize certain parts of the protein, these mutations, if they particularly uh, basically conform to the surrounding environments in a way. So conform to the surrounding environment means that, let's say in a hydrophobic environment, a hydrophobic to hydrophobic mutation, that will not have that much impact. But let's say in a if you consider the packing of the structure, so if closely in a tightly packed structure, uh, if uh, let's say an amino acid to amino acid mutation was observed that could alleviate the static hindrance in such a way, in some way, right? So basically, it covers the space in a proper way that no such static classes is observed. So these kind of mutations can also help in stabilization of these structures. In a way, we'll see that in a slide as well. I think I see another hand. Yeah, please.
Sir, I don't know whether I'm right or wrong,
Uh-huh.
but like the sir, it depends on the type of like right number of residues, right?
A number of
I
residues.
mean,
Many.
sir, I mean, if there are more charged residue in a protein,
Yeah.
then like the stabilizing amino acid would be a charged amino acid itself, right? Like, for example, if we take glutathione,
Hmm.
we don't see any hydrophobic residues.
Okay.
Then the stabilizing, like the major stabilizing amino acids are charged amino acids there.
So if you're
So,
talking about proteins, let's say some small residue spanning proteins or very small proteins that do not fold into a 3D, I mean that particularly do not fold due to hydrophobic driving force, then it could be possible. So if you're talking about some certain like small peptides or something like that, then it could be possible that they are mainly driven by charge-charge interactions, their kind of stability of those parts. But generally, if you talk about like larger proteins, let's say 200, 300, or let's say 1,000 amino acid residue spanning proteins, so of course there will be a lot of hydrophobic residue present, and they will tend to cluster together. So that is kind of in a standard sense for protein folding stability. I was kind of getting at this answer. But yeah, if you are talking about small, let's say peptides or something like that, then it could be possible that the stability of that peptide is driven by certain charge pair, charge interactions. That could be possible, yeah. So again, I have to look at your particular example. Then maybe I'll get to know about the structure of that one.
sir, I just had that in mind, so I asked.
Okay, sure. Okay. Yeah, it's interesting. Uh, so here again, accompanying this one, so I have certain a small part of a particular paper which I have referenced here. Now, what they have basically represented here is that physicochemical property along with the amino acid composition for halophilic, acidophilic, alkalophilic, thermophilic, cyclophilic, and their corresponding homologous normal protein. Okay, and this particular rectangle I have highlighted. So it is saying that it was observed that more than 80% of halophilic, thermophilic, alkalophilic, and acidic proteins, they showed a trend of having higher amount of glycine, alanine, isoleucine. Again, alkalines, hydrophilic, and halophilic proteins showed higher amount of isoleucine in their sequences. So since more than 80% of these proteins that they have observed, they sort of have this alanine isoleucine uh, based, basically, a trend of having higher quantity or higher amount of that. So I thought I would basically include this as part of this slide as well. So you can take a look at maybe this particular paper, which talks about the structural stability and so protein stability. So again, just accompanying that, I included one of the slides that I have also showed in some of the earlier classes. So basically the grouping of different amino acids into different basically subcategories, right? So for example, here we have aliphatic R group containing ones which are non-polar, then you have aromatic ones, then you have polar uncharged, then positive charge and negatively charged. So you can take a look at uh, this sort of table or this sort of characterization from the Leninger principles of biochemistry. Okay. Now, as we talked about the mutations in different regions, so different region means, let's say, the buried regions in the solvent exposed regions. So there, the favored mutations or the mutations that could stabilize the structures or destabilize the structures, the nature of that becomes different because it, of course, it of course relies on the surrounding environment and the nature of the surrounding environment as well, right? So in this particular case, it is asking about buried mutations. Now, buried mutations are basically meaning that it is occurring on residues which are in a buried position, deeply buried position. Deeply buried position will mean away from the solvent, basically in the core part of the protein fold, not solvent exposed, right? So those will be the buried residues. And if they are mutated, which of this property could be highly correlated with stability? So that means you have certain property which will correlate with the stability. So if that property the value of that particular parameter is low, then the stability will be low. If the value of the particular parameter is high, the stability will be high, and so on. So which one of these could be the answer here? Maybe highly correlates with stability.
Sir, hydrophobicity.
Yes. So since we are talking about bodied residues here, so they are not solvent exposed, and we know that these sort of regions are, of course, hydrophobic amino acid dominated, right? So in body mutations, of course, hydrophobicity would correlate with stability because let's say more hydrophobic nature or more hydrophobic residue that could increase the stability. And then if it's in a reverse way, if the hydrophobicity is lowered, or let's say it becomes a charged or polar type of mutation, then of course that will destabilize that part. Similarly, the opposite case will be again in the surface exposed or the solvent exposed residues which are present at surface, let's say. So those will mainly be polar or charged residue dominated, right? And if those sort of residues are to be mutated to a hydrophobic one, so they will also destabilize the structure in that part. So in that case, hydrophobicity will be inversely basically related with stability for surface mutations, right? Residues that are present at the surface. Now, again, in the similar sort of way, the question is asking which of these following mutations will be highly destabilizing in proteins. So you can imagine that if one mutation was from a residue to a different residue, which had basically the same type of nature, let's say their hydrophobic nature or the polarity and so on, if they had sort of same type of nature, then of course that will not have that much impact. That will not have that much impact in the structure of the protein. However, if the mutations are from one residue to a vastly different natured residue, then that would destabilize the structure of the protein, right? So in this, among let's all of this, which one do you think would be destabilize the cause of destabilization? So this is also related to one sort of table that I think Professor Gumiha Professor Gumiha shown in his classes, I guess. So here the actual answer was Y2A basically. So tyrosine to alanine. Now here in this table, in the accompanying table, if you notice, it is basically a chart, right? And the left hand side column basically represents the initial residue, so the non-mutated one, the initial starting point, and the first row, so this one, that is basically indicating the mutated residues. So the final point, if you say, and each of these boxes that we have, it sort of represents that residue to that particular residue. So this column wise, this residue one to this row wise, this residue two. So that change is being reflected by the values in the box. And what the values in the box basically indicate is that how many data points you have for these sort of mutations, and among them, how many stabilizing mutations do you have basically how many stabilizing mutations were observed. So the observed stabilizing mutations are uh, indicated in parentheses. So that means that outside of the parentheses, so those data points that have been observed, so they will kind of be destabilized, right? In a sense, destabilizing or neutral or also can happen here, but for the stabilizing mutations that are shown in the parentheses, uh, so from that we can infer, if we take a look at all of these sort of mutations. So Y2A mutation will be represented by this box, right? And then valin to histidine, I think should be this box, okay? And then E2M, so it will be this one, and then I2K, so it will be this one, right? So if we consider the inside values inside the parentheses as the stabilizing mutations, conversely the values outside the parentheses as destabilizing mutations, what we can see is that, of course, in the case of Y2A, this mutation, only one data point was observed where the mutation stabilized this part of the structure. However, other 22 mutations were observed which did not do that. If you compare that to, let's say, some other mutations that have been given in the options, we will see in this case parenthesis inside the parenthesis was 0, but we only had one data point for the outside parenthesis value, right? So you could say that that is not highly significant. For the other ones also, we do not have such highly significant value, but for E2M, we also see like one stabilizing mutations in this case as well, right? For Y2A, we have a lot of data, and among them, only one value or one observation was found, which would result in stabilizing mutations. The other 22 were not. So that is why in this case, Y2A mutation could be highly destabilizing in proteins. Okay.
So in exam, if such questions are given,
Mhm.
then do we have to remember the table or the table will be
Uh,
given?
no, no, no. I don't think you will have to remember the table. So in exams, the questions should be uh, basically such. I think the questions that will be given you can analyze using just the basic nature of the residues. For example, you could be given, let's say, a positive charge, negative charge mutation, and then you will be given a, for example, small hydrophobic residue mutation, these kind of things. So without even looking at any values, I think you will be able to compare the different sort of mutations there, which should be given in the exam. I think I see another hand in the chat.
Hello.
Yeah, yes, please.
Yeah. So is it that the aromatic are more stabilizing as compared to the other
Uh, Aromatics are not the most stabilizing, but uh,
aromatic
okay, so
responsible for more stability?
in this particular case, so Y2A mutation was considered, right? So if an aromatic Y was converted to a alanine, so
Yes.
that was highly destabilizing. But
Yes, but
yeah, yeah, yeah. But if you consider, like, for example, Y to any other amino acids, you will see different results, right? So in this Y column, you will see for the other set of mutations that are possible, those provide again slightly different results. So based on all of that, this particular
I'm
mutation was considered. Yeah, yeah, please.
yes, yes, yes. I got
Okay,
it.
yeah, yeah. And also it will depend on, let's say, the surrounding environment. So we do have a slide later on where how the immediate environment of the residues impact the stability in the sense of in cases of mutation, how that occurs. So we also have a sort of slide a little bit later, I think. So look at that also. Okay. So it also depends on
Okay.
this. It also depends on the size of the residues, how tightly packed the structure was at that particular region, what was the nature of the particular amino acid at that
Okay.
region, and so on. Yeah, yeah. Okay.
Okay. Okay.
Okay. Okay. So in the next question, it is asking again in a similar vein. So it is asking that a hydrophobic residues in a particular protein. So these residues are crowded in the core. So the core region, right? And alanine is mutated to valine, then what will happen to the structure? Now, in this particular case, so what they mean by the hydrophobic residues are crowded in the core. So they basically want to say that uh, the core region was very tightly packed. Okay. So it is a very tightly packed region of the hydrophobic environment, let's say the hydrophobic core. And in that tightly packed region or the crowded region, as they want to, as they went with the term here, so that tightly packed region, in that region, one alanine was mutated to a valine. Now, then what could happen in that mutation, following that mutation? So you have to remember that it was already structurally dense or densely packed, right? Yeah. So I think I see answers B and A also. Okay. So here you have to again take into consideration the size, right? The size of the residues. Okay. Now they are basically mentioning that it was already very tightly packed. So there is not a lot of, let's say, available free space for certain structural changes. And if in that uh, particular structure, if alanine was to be mutated to a valine, so if we see the side chain sort of structure of the alanine versus valine, we will notice that valine has a larger side chain, right? So in this case, what will happen is that if that mutation was to occur in a tightly packed space, so that will create steric hindrance because there is not that much space to accommodate the larger side chain of valine. Okay. So in this case, it will be creating kind of a steric clash, if you will. So here, let's say as visual representation, we have this alanine in this hydrophobic sort of cluster, right? And if that alanine was to change to a valine, which is a larger residue, so it will create steric hindrance. So that means in this case, the answer should be B. Now, similar type of mutations, whether they classify as stabilizing or destabilizing, that was discussed by Professor Gromiha. So I have taken that slide and put it here also, a similar sort of representation, right? So, for example, here what was happening in the first sort of mutation. So we had one negatively charged amino acid, we had one positively charged amino acid, and we can assume that they are forming charge-charge interactions, right? Which could be, of course, part of stabilization in certain regions of the protein. If this positive charged residue was to be mutated to a negative charged residue, of course, that interaction will no longer be there, the charge-charge interaction. Instead, it will get repelled by one another, right? So in that sense, that part of the structure would destabilize. So positive charge to negative charge, whenever there is this sort of change in the nature of the residues that are supposed to be present originally. So that could destabilize the structure, right? Now, what we saw in the earlier slide as well. So if we had the bottom basically mutation we are talking about, the bottommost one. So here we had this red residue, which was a smaller residue, but now it sort of mutated into a larger residue, this green one, right? Since there was no such space to accommodate the uh, the increase in size for the larger residue, so that was kind of destabilizing due to steric collision or steric hindrance, the part of the structure. However, if that structure was not tightly packed, but if that structure was loosely packed in its natural form, then what could happen is that if that loosely packed, in that loosely packed structure, if this red one was to be muted to a larger green one residue, so in that case, what could happen is that it could stabilize the structure, because the available space that was already there, it was sort of getting filled up by this large structure of the green residue, new green residue, right? And in that case, that would create like stronger hydrophobic interactions between residues, and that could help stabilize that part of the protein, basically. That could happen if that structure was loosely packed, meaning that there was already available space for that mutation to occur, where a larger residue could be accommodated. So it then can fill the space basically, and then create maybe stronger hydrophobic interactions and ultimately stabilize the part of the that particular region, the fold of that, or the uh, basically it could provide stability to that region, right? So as we see that it matters what type of residue is being changed to what type of residue, what is the surrounding environment of that region, whether that particular region was tightly packed, loosely packed, and so on, basically, whether we can accommodate for the extra structure, extra sizes, or whether it would create static classes and so on. So it depends on a lot of parameters, the stability of that part, and whether mutations, what kind of impact the mutations have on those regions. Okay. Now, based on that, what we talked about, so mutations providing the stability or destabilizing the structure, so we can convert that into some sort of free energy values, right? As we always quantify, let's say, change from one state to another state in terms of certain free energy values. So here also you can think of it, uh, think about quantifying that sort of mutations in with a free energy value, right? So that is what this question has done. So it is talking about 4 different alanine to valine mutations at different positions, and it has given the associated free energy value for those uh, particular mutations, right? For those different positions, and it is being asked that the most tightly packed residue position before mutation was which one? So before mutation, which of these residues was the most tightly packed alanine, basically? And here it has been given that positive value indicates stabilizing and negative value indicates destabilizing. So in some sort of other concepts, such as in free energies, in binding free energy calculation and so on, we thought about, what we learned was that the negative value would indicate like better binding, and the positive value would indicated loose binding, or the comparison of the binding affinity, right? So in that sort of way. But here the order is kind of reversed here. So the positive values indicate a stabilizing mutation, and the negative values indicate a destabilizing mutation. So based on that sort of sign convention, we have to consider uh, the calculation or the answer, basically. So first you have to identify, okay, since positive is stabilizing, negative is destabilizing. So which of these are showing stability, which of these are showing destability, and it is trying to ask what was the most tightly packed residue before mutation. So that means that after the mutation, if it was the most tightly packed, since the mutation is alanine to valine, again a small amino acid residue to a larger amino acid residue. So if it was already tightly packed, then after mutation it should create some steric hindrance, right? So in that sense, what could be the answer? I think I see answer D in the chat. So others maybe can also try to answer this one. So since it was already highly tightly packed, then of course the alanine to valine mutation will create steric hindrance. So that means it will destabilize that position, right? And then we can correlate that with the free energy values, which would give us the answer. So in this case, so this was the structure of alanine to valine. As I said, again, it is transformation or mutation from a small to a large amino acid, right? Uh, so in this case, the answer was A42. Now, why is that? Basically, we have to find out which of these, since it was already aligned into valine mutation, it was already tightly packed in that position. So which one of these mutations creates the most steric hindrance? So that will be, of course, which one of these positions had the most tightly was the most tightly packed, right? Because if it was the most tightly packed, then it would create the most steric hindrance. So that means which of these mutations is the most destabilizing. And here the negative values are being given as destabilizing. So then which one of these mutations has the most negative value basically? And if we compare, we see that A42 to V, it has a negative value of minus 2.4 kcal per mole if we compare with the other ones. So that means that in the 42nd position, since it was already highly tightly packed, the alanine to valine mutation that created a large, basically high steric hindrance, which means that that part of the structure was destabilized, which in this sign convention is the most negative value that is given. Yeah, I see answer in the chat C as well. So basically, yeah, this one, alanine 42. So it was the most tightly packed before mutation. Okay. So in the assignments or exams also, the sign convention I think should be provided. I think it will be provided whether the positive value indicates stabilizing, uh, basically stabilizing value, and then negative value indicates destabilizing, or it is vice versa in some other context. So I think those should be provided. So there should be not much problem regarding the sign convention. So we just talked about this in on other slide, right? So in which methods the mutants were divided into 380 potential substitutions, which if you remember, we talked about 380 beans, right? So the different set of substitutions or a particular pair of substitution was considered as a particular box or a particular bin where we can put all of the data points that we gather, right? And that was basically the average assignment method. Okay. So it was kind of the same question. So I think that was sort of the set of questions that I had. Some other questions that I that were kind of in the earlier classes, I also put them in the slide as well. So while talking about different interactions that can provide, let's say, stability or that play important part in binding affinity and so on. So one of the interactions was van der Waals interactions, right? So it is a crucial part of protein ligand interaction computing as well, where you have to sort of characterize how much of the binding free energy is being driven by the van der Waals interactions, and usually it is pretty high. So van der Waals interaction usually is like one of the dominant forces in protein ligand interactions. So the van der Waals interactions are basically identified as Lennard-Jones potential. So the equation for the Lennard-Jones potential are typically used for these calculations. Uh, and in this case, if you look at all of these options among these, so option A, which is the Aij divided by Rij whole to the power 12 minus the Bij divided by Rij whole to the power 6. So that will be the basically formula for computing the van der Waals interactions. Okay. So I think this was related to some of the earlier classes and so on. Okay. So for today's class, I think I will wrap up this one. So again, I guess we will meet at the next classes. Okay. So please make sure to join. Make sure to carefully go through Professor Pumiha's lectures, and then also try to join the other TA sessions that will also happen in Saturdays and Mondays and so on. And for the next set of lectures, we will focus on the other week's contents that Professor Kwame had discusses. Okay. Okay, I see. Yeah, yeah. The hand in the chat, please.
Sir,
Mhm.
I had a request.
Ah, yeah,
Uh,
sure.
can you can you give us a small projects or assignments that would help us in future, to understand better and to gain knowledge?
Okay. So, like, what sort of projects?
Sir, I mean, I'm a molecular biology integrated master student in molecular biology.
Okay.
So I'm planning to get into research, to join PhD. So I was hoping that understanding more about gaining deep knowledge in bioinformatics would help. So something.
So,
Uh,
okay. So, rather than me giving a particular project, so what you can do is that if you have so if you're trying to, let's say, learn the different skills on bioinformatics and so on, and the data analysis and so on. So what you can try to do is that you try to go through some papers, let's say recently published papers on certain topics that you're interested in, and in a lot of the papers, if they are like published at like high impact journals, so they also uh, give the associated codes and everything that you need to analyze the data. So what you could try to do is that what they have tried, let's say in generic bioinformatics analysis and so on, because in running, let's say, MD simulations and so on, you will need certain high performance computing facilities and such things. But in bioinformatics analysis, what you can do is that you can try to replicate some of the results in a much smaller scale that the papers were basically getting at. So let's say paper is trying to analyze different sequences and getting certain data from that. They're trying to arrive certain conclusions from that, derive certain conclusions from that. So you can go through that paper, what they are trying to say, what inference they kind of ultimately got out of their data and such things. And if they have associated codes, you can try to replicate the data and the observations that they have gotten for yourself at a smaller scale. You do not need to take all of the inputs or all of the data points that they have taken, but you can do it in a smaller scale. Okay. Uh, so in our lab, what kind of studies we do is basically uh, mainly based on simulation related studies, right? So we try to simulate different sort of biological macromolecules, let's say protein, let's say DNA, and so on, in different states, and then we try to infer some sort of observations that was not earlier found, let's say in other literature, right? So if some sort of mutations are causing uh, certain structural properties or dynamic properties to be changed, and such things. So also some virtual screening projects, and based on that, whether we can find novel ligands and such things. But those will usually require certain like powerful computing facilities. So I'm not sure if you will be able to do that. But on a smaller scale, you can try to do. So you can try to let's say find a protein first of all from PDB database, and then you can try to dock certain compounds, small molecular structures onto those proteins, and then compare the docking scores, and then try to see using different servers online whether those small molecules provide you with good aromatic properties or not, right? So these sort of analysis you can try to do, and then you will find you are basically trying to replicate a drug discovery in silico drug discovery project in that sense, right?
yeah, yes, sir.
So you can try to do that on a smaller scale. So whatever a certain paper has done, let's say they have taken a PDB structure, uh, and that they have done a virtual screening, and based on that they have gotten like, let's say, 10 or 20 hits, right? So you do not need to do the virtual screening, but what you can do is you can download the same PDB structure, and all the lead hit molecules that the researchers have found, the 20 hits they have found. So they probably will have they probably have also reported the associated pubchem IDs or some smiles, structural smiles, from where you can basically draw the structures, 3D structures, 2D structures, and so on, right? So they probably should have provided that. So based on that, you do not need to do the virtual screening, but you can do the docking of the 20 individual compounds with this protein, and then you can try to see, okay, if your result was matching with the researcher's results. and then you will see, okay, so my result is matching. And then let's say the better hits you are also putting through some online server such as admit lab and trying to identify the admit properties and so on. Then you will say, okay, I have kind of done this part of the paper, I have tried to replicate it, and it is agreeing with the researchers, right? So in that way you can devise certain small scale projects with already published papers if you are trying to learn these techniques like for these different domains. So you can try to do that. Similar things you can try to do with bioinformatics for sequence analysis and such things. If uh, you can try to see what the researchers were trying to get at, and then what kind of analysis they have done, and on a smaller scale, you just try to replicate that the same process basically. So a lot of paper also give associated codes, so that could be helpful. Uh,
Okay.
yeah.
So
Hmm.
the problem is I'm new to this like bioinformatics thing because
Hmm.
in my college it's mostly the course is oriented towards mainly wet lab techniques.
Okay.
So uh, I can I ask your help like if I have to do some troubleshooting like in
Sure,
the
sure, sure. You can ask me or you can ask the other TAs also. So if you need particular specific like part of some projects and so on, you can ask that. Uh, that's no problem. Yeah.
so can I get your mail ID so
Oh,
I could
sure,
chat.
sure.
So instead of using mail, I'll be using this Google Chat, so it will be more convenient.
Yeah, it's okay. So I have also shared the mail ID in the chat, so you can note it down. Uh, and then if you have any queries, then you can also ask me in the mail as well. Okay.
Okay, sir.
Yeah, yeah. Okay.
Sir, and also I had another query. Like, does your lab, I mean, Professor Gromiha's lab, offer any internships?
Uh,
Like maybe after getting some experience.
so I am actually not from Professor Gumiha's lab. Uh, I am currently working at IIT Indore basically. So at the Computational Biophysics lab at IIT Indore under Dr. Parimal Kar at BSB department here. So the other two TAs, they work under Professor Gumiha. Okay. For the internships, again, I think I remember for some of the last iterations of the courses. So what they did is that for this NPTEL course, the top scorers, I think they said that the top scorers will be able to do some sort of small projects or small internships with Professor Gomiha. So the top scorers for this particular bioinformatics course. I don't know if that is still the case for uh, this iteration of the course, but you can try to ask it in the forum maybe. Or if you particularly want to do project under Professor Gumiha, so I will suggest that you of course first ask the other TS because they are under Professor Gumiha himself. And then you can try to mail Sat himself also, right? Professor Gumiha, whether some sort of projects or internships will be available for you. So you can try to do that. I I work under Dr. Parimal Kar at IIT Indore. So again, if you are interested in project in this specific field, so particularly the macromolecular simulations and sort of these kind of things, so these fields, then you can try to mail maybe Dr. Parimalkar and try to get a response from that also. For Professor Gromiha, you can first ask, I guess, the other TAs, so they will be able to provide kind of a better answer uh, in that sense. Okay.
Okay, sir. Sir, I mean,
Uh,
although sometimes the interns are not usually accepted in few labs, so I asked you.
yeah, that's good. That's good. Actually, it depends on the guide, and then if there are already other interns that are working in the lab, so of course uh, they cannot focus on like a lot of interns, right? Because all of them will need guidance. So whether the availability of internship is there or not, you have to, I think, directly ask some of the lab members from that particular lab. So that will be, yeah. So in this case, since the Ramakrishna and Fatima, since they are under Professor Punika, you can try to ask them. I think.
Okay, so I don't like I have no preference as I'm learning. I just want to learn the skills. So I don't have any preference to particular lab. I'll check the papers and research that going on in your lab and also
Yeah, yeah, sure,
uh,
sure,
research lab.
sure, sure. You can
So
you
and
can
then
yeah. Mhm.
I'll try to contact you, sir. Okay,
Yeah, yeah, definitely, definitely.
thank you.
So, sir, you previously worked with Dr. Gromiha. I mean, how are you related to this course?
No, no, I have not worked under Professor Gomiha. So these particular live interaction sessions, so these live interaction sessions for the NPTEL courses, uh, so they usually kind of recruit the different PMRF scholars. Okay, so the PhD scholars who are getting fellowship under the PMRF scheme, the Prime Minister's Research Fellow scheme. So they are given like a chance to go for the TSS and taking the TSS under the NPTEL's umbrella basically. So for the PMRF courses, you have to complete a certain hours of teaching assistantship outside your home institute for a particular let's say year. Let's say in a year it is like around 52 hours. So NPTEL provides a platform for the PMRF scholars to be part of some courses as TA. So that's where basically I applied, and then since it's also helping me cover the total TA time duration, so it is quite good. So I also did TA for this course in an earlier iteration as well. And Professor Bromia students, of course, they are regular in this particular TA course. So I also applied and asked Professor Bromia whether I could, I could whether I would be able to perform TA in this or not. And Professor Bromia allowed me to basically do it.
So, for this, the one has to apply through NPTEL, and if selected, then approach the professor.
No, this particular TA session is offered only for the PMRF scholars. The
Yes, sir, I'm talking about PMRF only.
yeah. So usually when for a new NPTEL, let's say starting session, so they will provide all of the course lists, basically all of the courses that are available, and they will ask the scholars if you are interested in joining as a TA for this course, then you fill up the forms, you give your preferences, or alternatively, you can directly ask the supervisor of this course whether he will allow you or not, and that way basically they decide. So let's say for a certain course, if let's say 6 different PMRF scholars have applied for this course, then it is up to the instructor of this course, so or the supervisor in this case, or the instructor to allow the certain PMRF scholars. So they will look at your CV and everything, and then probably decide, okay, you are suited for this course, so you could help as a TA.
So, is it NPTEL approaches the scholar, or scholar has to approach when some opening is there?
No, NPTEL distributes the list for all of these courses to the scholars and ask them to fill forms whether they are interested in joining as a TA for certain courses or not. And then when the scholars fill the forms, then it is part of the then it is coming to the course instructor for that course to decide whether he will take those scholars as TA or not. So first NPTEL approaches the scholars, the scholar fills up the forms, and then professors decide whether these TAs will be suited or suitable or not.
So, sir, is it a paid assistantship, or
No,
it is only the part of NP this PMRF?
it is a part of PMRF, so it is not a paid assistantship. For PMRF, there is a strict criteria that you have to do outside TA outside of your own institute for 50 hours in a year. So that is a mandatory for PMRF basically fellowship. So you have to do that for each year essentially before your PMRF review for each year. So you have to do that in that time period. And NPTEL basically allows the scholars to join in this particular platform so that your the requirement of your T hours is also getting completed, and you are also helping as part of the basically you are conducting live sessions. So originally the idea was that interacting with let's say PhD scholars who are currently doing PhDs, the students can maybe get some insights into research and such things. So that's why NPTEL allowed to conduct these live interaction sessions, and they also give like completion certificate for how many hours you have done in this TA. So it is like helping both basically in that sense.
So this matlab one has to do this assistantship has to do for all the 5 years.
So if your PMRF is active, then you have to do the teaching assistantship. It is not that you can do it in NPTEL. You can contact other colleges also nearby. You can arrange online classes with other colleges also. You can conduct workshops also. So different types of way you can basically do TA for all of the required hours, the total hours. NPTEL is just they basically are
It's just
allowing.
a platform. Yeah, platform I got. So I means you can do virtual. I mean online also and physical classes. Either of the thing you can do. And I
Yes.
was just asking for the entire 5 years of PMRF. It needs to be done.
So it depends on for how much time your PMRF is active basically. So if
Generally, it is for 5 years. No? So
uh, basically after each year PMRF they conduct a review and then they decide let's say whether you will be a part of PMRF or not for the next one. So in that review basically you have to discuss your work, what you have accomplished in the past years, whether it is like matching up with the timeline that you have proposed earlier and so. So based on those kind of reviews, they decide whether your PMRF fellowship will be continued or not. So whether you will be part of the PMRF or not. So if you are part of the PMRF, if your PMRF fellowship is still active, so then you have to, of course, for the next set of uh, basically the next review period or the next time period, you have to conduct the TA sessions as well.
you told some 30 hours in a year.
No, it's I think it's around 50 hours in a year. 50 hours.
50.
Yeah.
So that 50 hours you can do in one semester also, or split also, or maybe two, three colleges like that.
Yeah. You
You
got
have to just complete that.
yes,
Okay,
you have to
thank you.
just complete that. Yeah. Yeah. Okay. So again, thank you all for joining the class. And
Sir,
yeah, please.
if you don't mind, I had another question.
Sure, please.
Uh, Sir, like, if you have no problem, can you just tell me about yourself, like your background on how you got into this PhD and about your work?
Yeah, so basically so I'm from West Bengal and I did my bachelor's in physiology actually. So BSc in physiology honors from Vidyasagar University at West Bengal. And then after that, through IIT JAM, I basically enrolled here at IIT Indore. So in the MSc Biotechnology program here in the Department of Biosciences and Biomedical Engineering. So during the MSc, the 2 year duration of the MSc, so at IIT Indore, and I think for most IITs and ISRs, they also follow this. So what they do is that the second year of that course will be a project-based course. So you have to basically join one of the labs at the department, and then you have to do project under the supervisor basically. So I also joined under Dr. Porimalkar here at the computational biophysics lab because I was at the time interested in these computational fields, the molecular simulations, virtual screening, docking, drug discovery, and so on. So I joined Dr. Kar's lab here. I completed that project, and then after that, so for MSCs here, uh, they basically allow a conversion. So if you have certain criteria fulfilled, so you will be allowed to convert from let's say MSc to PhD. Okay. So that is kind of a part of the dual degree program here, as they call it. So MSc plus PhD dual degree program. So I also applied for that and I got the dual degree like approved. So then I continued in the same lab here under Dr. Porimalkar in the computational biophysics lab for PhD. Uh, and what the PhD project I'm doing in it is basically so in our lab, as I said, so we mainly focus on like biomacromolecules and their simulations and what sort of different observations and inferences we can get from that type. So we focus on conformational dynamics of proteins, protein DNA binding, protein drug binding, protein glycan binding. So they help in like viral signaling process and like in viral infections, the signaling goes down and the glycans also play a role in that sugars. So in these sort of systems and their simulations, we do all atom simulations. we do biased simulations, Gaussian accelerated simulations. We are trying to do Markov state models and so on to basically infer the internal dynamics and then the time how much one state to another state change basically it can take. So these sort of things we basically try to analyze. In my case, I am trying to uh, I'm basically working on a protein which is called spleen tyrosine kinase. So it is one protein that is heavily involved in autoimmune disorders and also in different other diseases as well, cancers, neurodegenerative disorders. In those sort of diseases, this particular protein is also playing a good part. But in its classical signaling, it is basically a part of the autoimmune disease. So it helps in proliferation of B cells, B cell maturation, and so on. And basically, in autoimmune conditions, so against SOIK, there is already one drug. It's called fostamatinib. So basically, what is happening that this drug will occupy the ATP binding site of the spleen tyrosine kinase, and then it will block the downstream signaling, which helps relieve the symptoms of the autoimmune disorders because this signaling is playing a part of the B cell maturation and such things. So it helps relieve that. So I'm trying to uh, basically my project is having different parts, you can say. So firstly, to identify and optimize new drugs because already available ones or the already tested ones which are currently under process, they sort of have different side effects and so on. And if we can make a new drug which will not only have less side effects but could have better affinity towards the particular protein, let's say better selectivity towards this protein, then we could basically try to propose that as a novel candidate, right? So one of the parts of my project is regarding that, basically identifying and optimizing novel drug candidates against this particular protein. And then other parts are basically related to conformational dynamics of this protein in different states. For example, what kind of changes occur in a phosphorylated state versus a non-phosphorylated state in that proteins, because we know that phosphorylation in proteins basically drive conformational changes. Certain conformational changes which can lead them towards, which can basically help the downstream signaling to progress, right? So what sort of state changes are being brought about by these different kind of post-translational modifications like phosphorylations and so on. What could be the impact of mutations on these proteins? We already have, let's say, uh, in literature, previous literature, different mutations have been reported, right? So what sort of changes are these mutations bringing to the protein and the overall protein dynamics, the conformational dynamics? So these sort of analysis we basically try to do in our lab. So protein conformational dynamics, and then protein glycan interaction, protein drug binding, and so on. And if we talk about the tools that we use, so usually for something like virtual screening, there is a lot of software suites, right? There is Schrodinger present, that is widely used. There is MallSoft present. So these sort of kind of software suites we use whenever we have the ability to do. For the simulations, we use particularly AMBER. So it is a again simulation suite that has a lot of different parts. So it has it's like very good and uh, basically well collected set of software packages, right? You can say, right? So Amber we use for simulations. It has protocols for different kind of things, all atom simulations, biased simulations, and so on. Uh, if you do not have access to Amber, you can try to learn Glomax. A lot of Glomax is also free. And then again, for the other set of analysis, we basically use Python and the different libraries of that, right? So for analyzing the data, the output, uh, and then representing them in different formats. So you can use different Python libraries, first of all, NumPy, Seaborn, Pandas, Matplotlib for plotting, and different things. For other molecular dynamic trajectory analysis, also you can use Python, like MDTraj library and so on. So yeah, basically these kind of software suites, Python for data analysis, and then we try to just interpret the data as we get it.
Yes, sir.
Yeah, yeah.
Sir, can you share your PI's like name or
Yeah.
uh,
So I'll share. Okay. So I'll share the web page for our group in the chat, so you can take a look at this link. So my PI is Dr. Parimal Kar at the Department of Biosciences and Biomedical Engineering, or BSP at IIT Indore. Okay, so you can go through that link and then you can look through different tabs and so on, and then you will get an idea basically.
Okay, sir.
Yeah, yeah,
Thank you, sir.
yeah, yeah. And again
Thank
for
you very much.
yeah, and again particularly for Professor Bhumiha and the projects like related to his lab, so you can ask the other TAs basically, right? Ramakrishna, Fatima, they will be able to better answer those things. Okay. Okay. So
Okay,
again,
sir.
yeah. So thank you again for joining, and then we'll meet at the next class. Okay. Thank you.